I'm going to talk about a collaborative project with Bruker Cellular Analysis. The title is Development of an all-chip binding assay to select clones producing high-quality bispecific antibodies. My name is Shu Wang, and I'm a Principal Investigator at Incyte. I do cellular development to produce monoclonal and bispecific antibodies. For more information, please check my file. Let's go to the outline. I will present rationales for developing an all-chip binding assay, design and validation of the all-chip binding assay, test clones selected with the all-chip binding assay, and summary. Let's go to the first part. Rationale for developing an all-chip binding assay. This slide shows an overview of the cell line development process. It includes transfection, mini-pool and bulk pool development, single cell cloning, cell expression and fed batch evaluation, top 6 clone selection, and RCB generation. Then we will perform stability study, and upstream we will perform platform fit study. Based on the data, we will perform lead clone selection, MCB generation, and MCB testing. Then the MCB will be released based on the testing results. For single cell cloning, we use a beacon instrument. The quality screens are typically done close to RCB generation. We always get a lot of bad quality clones. It's better to this screen in the earliest time. The all-chip binding assay makes this possible. I will talk about this later. This slide shows the cell line development of the beacon instrument. We will load single cells from stable pool into nanopore chambers. Culture cells to measure growth characteristics. Assay colonies for productivity and quality. Recover colonies in multiple well plates for scale up. The cell line development process to produce monoclonal antibody is relatively simple, and the process to produce bispecific antibody is more complicated because the bispecific antibody is heterogeneous. At the end of fed batch evaluation for clone development, we submit pre-purified samples to perform quality analysis. According to the results, we can find good clones with a high percentage of heterodimer. We also get bad clones with either high percentage of arm A antibody or arm B antibody. Expand the heterodimer. To prevent the bad clones from cell expression and fed batch evaluation, we developed an octet binding assay. First, we use p-stacked ligand A and ligand B to make standard curves. Then we measure arm A and arm B antibodies concentration according to the standard curves. Then we will calculate arm A to arm B ratio and select good clones. With this octet binding assay, we can exclude many bad quality clones. However, those bad quality clones are already exported from Beacon. We should prevent those bad clones from Beacon export and further evaluation. Then we can evaluate more good quality clones and increase the efficiency of cell line development. Therefore, we decided to collaborate with Bruker scientists to develop an all-chip binding assay to achieve this goal. This part, I will talk about design and validation of the all-chip binding assay. This slide shows the design of the all-chip binding assay. We will label ligand A and B with Alexa 594. Perform assays with labeled ligand A, ligand B, and spotlight hue FC sequentially. Detect fluorescence intensity and calculate antibody concentration. Determine stoichiometric ratio of arm A to FC and arm B to FC. Validate the assay with clones, mini pools, and bulk pools. The quantity data of those clones, mini pools, and bulk pools is already known. Then we can calculate the actual arm A to FC and arm B to FC ratio and compare the ratio to the all-chip ratio to validate the assay. This slide shows the clones and pools for all-chip binding assay validation. We have a lot of clones, mini pools, and bulk pools. The product quality varies. The heterodimer percentage rate is very wide. Different color represent different rates. I want to point out that we have two bulk pools, TF4 and TF3. They all express single arm antibodies. TF4 express arm A antibody, and TF3 express arm B antibody. The TF4 can be used as a positive control for the ligand A assay and a negative control for ligand B assay. Conversely, TF3 can be used as a positive control for ligand B assay and a negative control for ligand A assay. As I mentioned earlier, we use ligand A and ligand B assay to measure arm A and arm B concentration. So we performed the assays and find that FC and arm A assay scores are linearly correlated with feedback titer, while arm B not detected. This picture shows assay images of spotlight hue FC and Alexa 594 labeled ligand A. We couldn't detect Alexa 594 labeled ligand B. We did some experiments to find the root cause. There's some reasons. First, the ligand B is not labeled. Second, the labeled ligand B cannot bind to the antibody because the structure is disrupted. Third, there is an antibody ligand aggregation. We use a labeled ligand B to stay cells expressing IMPD or the bispecific antibodies. We found there is a staining. This indicates that ligand B is labeled and it can bind to the antibody. Also, the binding is confirmed with the octet binding assay. Then we performed aggregation assay with cyziclusan HPLC. We found that the bispecific antibody aggregates after binding to the ligand B. We believe the aggregation issue is a reason for poor ligand B signal. Because we cannot use the ligand B in the assay, we use the two reagents, Spotlight QFC and Alexa 594 labeled ligand A to perform assays in two bulk pools, TF3 and TF4, and two cell lines, 2C11 and 4F7. Those two cell lines express the bispecific antibodies, and TF3 express IMB antibody, and TF4 expresses MA antibody. The titer of TF3, TF4, and the two clone, 2C11 and 4F7 is already known. I also want to point out that to compare the ligand A score of TF4 with the titer, the score should be divided by 2 because the TF4 expresses single arm antibodies. Because FC and RMA assay scores are linearly correlated with the fat batch titer, we decided to expand the assays to other clones, mini pools, and bulk pools. We found that chip stoichiometries correlate well with off-chip data. An initial assessment of the bispecific heterodimer percentage assay showed excellent agreement between on-chip and off-chip data. The R-squared value is 0.89. Further improvements were made to the assay calibration method to further improve the consistency of the visometric measurement. After the assay is validated with the clones, mini pools, and bulk pools, we decided to use this assay in our cell line development process to select clones expressing high quantity bispecific antibodies. This part I will talk about testing clones selected with all-chip binding assay. This slide shows clone selection experiments from bulk pool TF7. The goal is to select for best quality and the highest titer clones out of bulk pool TF7. We select 96 clones out of the pool. We select 3 groups. Good clones indicated by the gray red rectangle. Bad clones indicated by the 2 red rectangles. Clones with lower titer indicated by the purple rectangle. The criteria for best quality is defined as MA to FC ratio between port 4 and port 6. The criteria come from our historical data. We have 3 clones with quantity data. We calculate the MA to FC ratio and plot this ratio to heterodimer percentage, we found that 19 of 21 clones with this port 4 to port 6 bond have the best product quality. The two clones within this ratio also has a heterodimer close to 80%. This result indicates that port 4 to port 6 bonds cover most good clones, and we can use this bond to select good quality clones. This slide shows the clones evaluated in fed batch assay. In total, 42 clones were evaluated in standard fed batch assay. We first tested 7 clones in tube spin bioreactors, we call them TPP, for pilot validation. Those 3 clones are from the 3 groups. We found that the off-chip and on-chip ratio correlated very well, and the selected lower clone, the clone with lower titer has low fed batch titer. This indicates that the assay is valid, and we performed further validation in Amber 15 for 35 clones. Of the total 42 clones, 36 with CEX HPLC data to calculate actual RMA to FC ratio. We couldn't get enough material for quality assays for the rest of 6 clones because the titer is too low. This slide shows that highly producing clones are selected. 35 clones from Amber 15 fed batch assay are analyzed, and the titer is normalized to bulk pool titer. The bulk pool is a mixture of clones. 50% clones have titer higher than the bulk pool, and the rest of 50% clones have titer lower than the bulk pool. In our experiments, only 3 selected good clones have a lower titer than the bulk pool. It's less than 10%. We also have 10 clones. The clone titer, bulk pool titer ratio is over 3. This result indicates that the assay is efficient in selecting high titer clones. We also find that the assay is more efficient in selecting clone product quality. As I mentioned, we have 36 clones with CX HPLC data, 9 with good ratio between port 4 and port 6, 26 with bad ratio. Among them, 21 with high ratio over port 6, and 5 with low ratio below port 4. 1 clone was excluded due to lower limit of detection by the assay analyzer software. We found that 9 selected good clones have the best product quality. We also found that 9 selected bad clones have high product quality. Therefore, the false negatives, and the false negative rate is high. It's close to 35%. To solve this problem, we did an analysis to minimize false negative hit rate by design. The port 4 to port 6 bolts were designed to minimize false positive, allowing for false negative. Alternate approaches include expand the bolt to reduce false negative, allowing for false positive. When we expand the bolt, we can reduce false negative rates. And when we further increase the bolt, we have higher false positive rates. Therefore, we decided to use port 3 to port 7 bolts to analyze the data. We found that expanding the bolt reduces the rate of false negatives. Before we expand the bolt, we have 9 true positive clones and 9 false negative clones. After we expand the bolt to port 3 to port 7, we have 15 true positive clones and 3 false negative clones. The false negative rate is greatly reduced. We decided to use port 3 to port 7 bolts for future project. This is a summary of this presentation. One labeled ligand and spotlight hue Fc are enough to detect antibody quality. In our case, the labeled ligand B doesn't work. But it's better to have 2 labeled ligands. The assay data can support each other, and the assay data will be more accurate and the selection will be more powerful. Observed cheap stoichiometries correlate well with the actual MA to Fc ratio. The clones, ini pools, and bulk pools validate the all-chip binding assay. All-chip binding assay increases bispecific cell line development efficiency by selecting best quality clones from Beaker. Expanding the bolt decreases false negative rates. This is the last slide of my presentation. I want to thank Jennifer Hultman, Leibin Wang, Chenxin Zheng, Qingxiang Wei, Oscar Lara Velasco from Incyte, and Philip Elms, Gadis, Eric Sackman, Aurora Fabry Wood from Bruker. I also want to thank the organizer to invite me presenting my data. Lastly, I want to thank the audience. I will take the questions later. Thank you.